Hi, this is Miss Litton, and this is a 7th period review to get us ready for our final for AP Biology Unit 1, and these are my participants. Say hi.
Hi.
Okay, so I want to be efficient with our time, and I'm not calling you out in any way. Some of you may have started your review for final process. Some do it as you go along, and you make sure that you're never behind. You're always constantly going back and reviewing. Some of you haven't even thought about the final until today, our first review. Some of you are at home right now eating toast, not even coming. But wherever you are on the spectrum, let's talk about a few things. We have had a review over Unit 1 already. Is that accurate?
Yes.
And that is already on YouTube. So you could go back and look at that. We have already discussed the essay questions. So you could go back and watch that and refresh your brain about that. What I'm here to do is just kind of point out some main points to you, for especially those who haven't started yet, and those of you who have, kind of confirm what you have reviewed already. So let's break it down. When we come to something that seems difficult, more difficult than others, then we'll flesh it out a little bit more. But keep in mind, there's a review already in place for all 4 chapters in Unit 1 and essays. Go back and look at that. That will have more of the details that you might need. Okay? So remember, there's 2 aspects that you are going to be reviewing for the multiple choice and reviewing for the essay portion of your final. Now, there can be some new types of questions that I ask on both the essay and on the multiple choice that you couldn't have answered in isolation of a single unit. But now that we have so many chapters underneath our belt and so many topics under our belt, the questions can be bigger, right? Because I can span multiple units because I know what you know. So don't be surprised on the essay portion if you go, wait, what unit is this? It could fall over 2 or 3 different units. So just be aware of that. Another thing I would highly recommend as you're starting to prep for your final is your book has so many resources. Practice tests, practice essays. If you look again at the beginning of each chapter, okay, like I'm looking at chapter 3 right here, it says the chemistry of organic molecules, and it breaks it down into 5 different topics. Remember, these 5 different topics are the same way I broke down your notes. So anything that's scary when you look through these topics, you can go to 3.2 in your group shared notes and you will find the information to the level that I expect you to understand it. Also, on each of these chapters, it says before you begin, take a few moments to review the following. That's really a good idea. As you're progressing through each chapter, make sure you can answer those questions before you start reviewing that chapter. The other thing I would look at is these big ideas, because when ultimately we're trying to get you ready for the AP exam, not just this final, right? And so you have to dock all of this information and weave it into each of those big ideas. Remember, the first big idea was on evolution and how that drives both diversity and unity, right? That was big idea number 1. And big idea 2 was about energy, right? Am I getting it right? Big idea 2 was about energy. Big idea 3 was DNA storage, transmission, and response. Is that right? And then the last one was interdependent relationships. So you want to look to see, how does this topic fit in? And they have statements at the bottom where it shows big idea 1, the diversity of biological life is a result of changes in DNA sequences and the biomolecules for which they code. So it's going to take this chapter for you, and it's already telling you how it fits into those big ideas. Some of you get so lost in memorizing the details of a single factoid that you can't answer a general question that is easy. And I'm not saying that to put you down. It's the way you've been trained your whole life in schools to memorize all these factoids, right? But you have to be able to synthesize this into a bigger concept, and I want to encourage you to do that by looking at the big ideas. The other thing I would do when you're trying to like approach a review for a final is I would take the chapter and just literally go through each page. And just like we do in class when I say, okay, blue, explain this, slate, explain this. Try, it'd be great if you worked with a great study team, but I know some of you are lone rangers and you feel best when you're working alone, and that's fine. But go through these different pictures and go, can I explain this diagram? Do I understand what this is about? And chances are, if they have a picture to correlate with the reading, then it must be a big ticket item, right? And so that can help narrow it down so you don't get lost in everything. So just go through. Start with what this chapter's about, see if you can do what it says at the beginning, see if you can tie it into the big ideas. Go through each page, look at each picture, see if you can understand each picture. Then I would read through your group shared notes on that. Skim over the stuff you know well. Just, go through it. And then your next thing you want to do always is answer the questions in the back. And they're giving you practice questions that are multiple choice based. They're giving you, they review the big ideas in the back. There's a summary. You might not have time to read all those summaries. You've got a lot to do in the next 3 weeks, so I understand that. But that on a chapter that's harder for you, go through and read that summary and then go and take their test and respond, especially to the ones where you apply the big ideas, because that's what you're ready to do now. Okay, and look through those and apply the science practices. Do the thinking critically. Go onto the book and answer even more questions. And just don't get lost in the minutia of details. There will be questions like that, but you'll be surprised how many of them just depend on your working knowledge of the 4 big ideas and the science practices. The other items I would do is I have given you Bozeman's all along the way, and you're on the honor system for watching those, and some of you are #hideous about watching that. So I may change that second semester and start mandating some of those, because some of you are not being responsible in watching those. Remember, I'm having you watch those ahead of time to prepare your brain. At this point, you can't do anything about whether you invested that time or not, but you can use it to review. And there are 55 big ticket concepts you need to know, and he has 55 videos to go along with it. And you will have watched through all of them if you follow my watching plan, and you will also do the supporting ones. If not, you can go directly to Bozeman, look at the link, and go through and start watching each one as you go through, or go to the ones that I told you to watch and watch each one of them through. Okay, these are all things that you could have done as you went along. Take a moment right now and assess how well you've been keeping up with the material and how well you've been reviewing the material, and just share that with somebody next to you. Be honest, don't lie. Now hear me. After you take finals, it's going to be December winter break, which most of you really, really need. And you're going to come back in January. It's going to be a whole new year, right? 2017. We're going to have January, February, March, April. 4 months. And then it's going to be May. And in May, we're starting to take that AP exam. How you feel in May? Let's just fast forward, 4.5 months here. How you feel in May about yourself and your preparation is going to be largely dependent on what you do in the next 2 weeks. Because when I start talking to you at the in the middle of the semester, and we start getting closer and we start doing more and more prep specifically for the AP exam, you're going to wish that you did a really good job right now with everything that you have. You want to be a master of the material. You want to have a working knowledge of it. You want to be able to support your points. You want to claim evidence, reason up the yin-yang on every statement that you make. So you're going to be grateful in May for that. You're going to be grateful as you get ready to prepare that test. And the more times you force yourself to take assessments, like how many of you are taking advantage of the online textbook multiple choice? The more time you force yourself to take those assessments and see a score and watch your improvement, the more confidence you will have when you go to take a test with me. Okay? So build your biological confidence by doing a great job. This is not the time to have drama in your family. This is not the time to break up with a boyfriend or girlfriend. Okay? You need to break up the first day of winter break. Okay? And then you have all winter break to recover. Don't do it right now. If you've got, you might all have a friend right now who's one of those drama friends, and as the stress ramps up for finals, they create a whole new scenario of problems that they start spinning. And the more smiling poo you spin as an individual, realize eventually you're going to have to lick the spoon. So don't be that drama person. Just stay focused, get enough sleep. You don't want to get ill. Keep your exercise, healthy food, drink water. These are things that are going to pay off for you, not only just for my final, but all your finals. Okay? So those are my words of encouragement from watching kids year after year after year take finals. So let's discuss chapter 1, which is probably the easiest, or unit 1, of all your units, which is nice. Okay, so the first one on A View to Life, we went over characteristics of life. Let's do it really quick. Living things are organized, made of cells. They acquire materials and energy. They maintain homeostasis. They respond to stimuli and reproduce. They grow and develop, and they adapt and die. I want you to think about those words. You learned all of those things. Living things are organized. How are they organized? Atom, molecule, organelle, cell, tissue, organ system, organism, population, community, ecosystem, biosphere. Now think about that. Right now, that very first statement, living things are organized, you ought to understand atoms, the atom story. Atoms are made of protons, neutrons, and electrons. The protons and the neutrons are in the nucleus. The electrons are in energy levels 2, 8, 8 to 32. 8, 8. 8 is great. Octet rule. You ought to understand atoms and how they are built. Atoms build molecules. There are 3 important inorganic molecules. What are they?
Carbon dioxide,
water, and oxygen. There are 4 important organic molecules. What are they? Lipids, proteins, nucleic acids, carbohydrates. You should know those are all polymers. What are the monomers for those? Carbohydrates are built out of monosaccharides. What do we call those? Sugars. Do you remember the 3 monosaccharides? Glucose.
galactose?
No, it's disaccharide.
Glucose.
Fructose. Galactose.
I was not right. Okay, you were right.
Okay?
What a joke.
Then we have some disaccharides, right? Maltose is glucose and glucose. Sucrose, table sugar is glucose and
Sucrose.
sucrose is glucose and fructose. And lactose is glucose plus galactose. Then we had carbohydrates that were for energy.
I know these.
Say them.
Starch.
Starch
Glycogen.
and glycogen. Who makes starch?
Plants.
Plants. Who makes glycogen? Animals. We need sugars in order to maintain homeostasis by doing what? Cellular respiration, of which you know all the steps, right? Maintain homeostasis. We do cellular respiration. Living things acquire materials. What is that process by which they acquire it? Autotrophs. What do they do? Photosynthesis. You see what I'm saying? those simple words with the lame little song we had at the beginning, you could write books on now for me, right? So back to those important molecules. We discussed carbohydrates, their energy importance, but they also had structural importance. What structural importance did they have? Cellulose, cell wall, what else? Chitin, right? So those were carbohydrates. Then we talked about proteins. What are proteins built out of?
Amino acids.
Amino acids. How many different amino acids are there? 20. Do you know how to stitch amino acids to build proteins?
Yes.
Yes, through the process of what? No. What's the process called? Transcription and then translation. Right? So when we talk about how you make proteins, we can tell you so much about how to make proteins. We can talk about the types of proteins just in a cell membrane. There are what? Channels. There are carriers. There are receptors. There are enzymatic. Oh wait, did we do cell recognition? And then there are junctions. Okay, so those are just types of proteins you would find in a cell membrane. What's a cell membrane there? To decide what goes in and out. So we could talk about 3 ways to cross a cell membrane. We could talk about what? We could talk about through the phospholipid bilayer using a channel or a carrier and whole membrane, right? Endo and exocytosis. Do you see how I'm connecting this for you? Don't make anything in isolation anymore. So there's diffusion through the phospholipid bilayer, channels and carriers, and whole membrane, endo and exocytosis. Was that what you were asking your neighbor for? Oh, that's what I thought. Okay, so then we talked about proteins. When we talk about proteins, we could talk about how proteins are made. Who codes for those proteins? DNA. You could tell me right there, that's one of the 4 important organic molecules, so I'll just pause on that for just a minute. But yes, you are right. You hook proteins together through what process? Dehydration synthesis, and that's occurring on the workbench for protein synthesis, which is a ribosome. What kind of bonds do you form? Peptide bond, which is a type of what kind of bond? Covalent bond. A covalent bond is when you share electrons. If you share them fair, it is called a nonpolar. If you share an unfair, it's called a polar covalent bond. Now, proteins, when you put them together, we have chaperone proteins who have to fold them correctly underneath the what kind of control? Post-translational control. And we know that folding, we're taking the primary structure, and then you have the secondary structure, which either can be a what? Alpha helix or a beta sheet, right? And then that is not dependent on the R groups, but the tertiary structure is, right? Because those R groups could be charged, not charged, hydrophilic, hydrophobic. And the tertiary structure, remember, is the folding pattern imposed upon the secondary. And then the quaternary structure is if you have more than one chain. Okay, so we're talking now about proteins across 3 different units, right? Okay, proteins. Let's talk about lipids. What are the 2 categories of lipids? What?
Steroids.
Fats and steroids. And fats are made out of what? What are fats made out of? Fatty acid chains and glycerol. And those fatty acid chains could either be or saturated or unsaturated, depending on the number of hydrogens. Yeah, and we talked about some of them being healthy for us and some of us not being so healthy. What were the unhealthy ones? Saturated ones, which normally comes from, and it is normally at room temperature. Good job. Exception palm oil. Okay, and then we talked about steroids. What are steroids made out of? 4 carbon rings. Good, hooked together. Where might I find a steroid in a membrane? Cholesterol. Right? He's like, boom, shagalaga. And this affects the fluidity of the membrane, right? If it's really warm, it helps keep them what? Together. And if it's cold, it keeps it from freezing, right? Okay, and then we talked about nucleic acids. Boy, did we talk about nucleic acids. Nucleic acids are built out of nucleotides. And you could tell me the difference between an RNA nucleotide and a DNA nucleotide, right? You could tell me about RNA, instead of being double-stranded, and it has the sugar instead of, and has the base. And you could talk to me about purines and pyrimidines and number of hydrogen bonds. You could tell me the central dogma. You could write an essay on the central dogma. Okay, because central dogma is DNA makes more DNA through replication, which you could tell me about. The DNA what? Unwinds and unzips, helicase, and then we have to put down some what? SSB to keep them apart, and then we need some primers, right? One primer on each of the leading strand, multiple primers on the, and that's because enzymes, we know, are very what? Specific, and they can only read from the 3 to 5, making complementary 5 to 3. And we use DNA polymerase 3 to work off of those primers. That's an example of an enzyme, which is an important molecule, which is a protein, right? And it reads 3 to 5, making complementary 5 to 3. Then we have to use DNA polymerase 1 to remove the primer, right? And then we use ligase to seal them up. And then these are now what? Sister chromatids held together by a centromere that separates during anaphase, if we're doing the cell cycle correctly, which makes you think about regulation of the cell cycle. Okay? We can go anywhere. You can link any story together of your biological molecules. Okay? So that took care of
Every chapter.
every chapter. Okay, done. Hence biology. Okay. So when we talk about, if we go through living things are organized, made of cells. Okay, so that's right here, cell structure and function. You could tell me about the organelles in every single cell and the role it plays in maintaining homeostasis, or the role it plays in proteins and how you make or modify them, right? You could talk about the stomach of the cell and how its role is in degrading and breaking down things, or how it contributes to apoptosis. Maybe the P53 gene sets it off and caspase in order to degrade a cell that's not working properly anymore. Remember tumor suppressor genes? Remember proto-oncogenes that can become oncogenes, right? You could talk to me about the ER, and now you can talk about what makes the ER rough is the what on them? Where you're making proteins, and you know how to make proteins, how they move into the ER, and then they move to the what? Golgi, and then they could go and get secreted by the cell, or they could stay within the cell, right? You could talk about, what else? The centrioles and their role in cell division, the things that look like churros. Who doesn't have a centriole? Plants don't. But they have a microtubule organizing center, right? That shoots out those radiating microtubules. And we could talk about how centrioles kind of look like basal bodies that anchor cilia and flagella, right? To help move a cell around. Remember the 9 groups of 3 microtubules? 9 groups of 2 with 2 in the center? Okay, so all of that we can review then, the structure of the cell. Living things are organized, made of cells. They acquire materials. We talked about photosynthesis and energy. They maintain homeostasis. We've talked a little bit about that. They respond to stimuli. That's coming up still when we talk about nerves and cell-to-cell interaction. Living things adapt. Boy, we could talk all about adaptations, couldn't we? And population genetics. Evolution is change over time. You could talk about the history of evolution. You can compare Lamarck and Darwin. You could talk about the things that disrupt the Hardy-Weinberg equilibrium. And then if we could maintain the Hardy-Weinberg equilibrium, then no evolution would occur, right? All of that could be compared and contrasted. And as we move into the next chapter, we can talk about macroevolution as well. Okay, so the next thing in chapter 1 we talked about was the big ideas of evolution. If you look back over those will almost be comical review for what you now know from what we first talked about. Big idea 2 was energy and molecular building blocks. You know metabolism, you know all about energy. Information storage and transmission. Right now you know about DNA and how it codes for proteins. We'll talk about other information and behavior later. And interdependent relationships, we're going to get more into that in some later chapters, but we do know the organization of life. Science practices. You know science practices are going to be on your AP exam, right? And every chapter has science practice questions where you have to evaluate a graph or analyze some data, and I encourage you to do that. Chapter 2, basic chemistry. We have already reviewed the atoms and electrons. We talked about covalent bonding. What are some other bondings we could have discussed? Ionic bonds, and that's when you gain or lose electron, and then you're either, an ionic bond is when you're attracted, right? Chemistry of water, properties of water. This relates well to polar covalent bonds, right? Because oxygen hangs on to the electrons a little bit tighter from hydrogen, then oxygen is slightly negative, the hydrogens are slightly what? Positive. So between water molecules, the slightly negative oxygen is attracted to the slightly positive hydrogen of a different water molecule, yes? And that's the hydrogen bond. And we know it's hydrogen bonds that holds what? DNA together. How many hydrogen bonds between adenine and thymine? And between guanine and cytosine? And this is the tertiary structure we're talking about in the folding pattern of proteins, oftentimes related to the hydrogen bonds. We're mostly made out of water, and that's full of hydrogen bonds. And we talked about high heat capacity, right? High boiling point, remember? And we talked about the cooling properties of water. It can fill a vessel. We talked about water tension. We talked about frozen water being less dense and then it protecting the life underneath. So all of that relates to the properties of water and the biological benefits. And we talked about acids and bases. Acids are a measure of the amount of what? Hydrogen ions, the pH, good. Then 4 important organic molecules. We have already just reviewed the 4 important organic molecules just now. That was chapter 3. And chapter 4 takes us to the cell theory and cell structure. One thing we didn't mention earlier was comparing prokaryotic and eukaryotic cells. Which one is smaller? Prokaryotic. Yeah, which one would you consider simpler? Prokaryotic. Yeah, and things now that you understand, we know as far as DNA goes, who has all of these introns in it, prokaryotic or eukaryotic cells? Eukaryotic. Which ones? Eukaryotic, good. We also know that 3 of the 5 control mechanisms take place in a nucleus. Prokaryotic cells don't have a nucleus. What are the 3 that take place inside of a nucleus? Chromatin structure, like all your epigenetics. Transcription and post-transcription. Translational and post-translational take place outside. They don't have to worry about genetic junk. They don't have to worry about unwinding DNA. Remember, theirs is mostly, can RNA polymerase get on the promoter? It's mostly about negative control and repressors, right? And then as soon as the mRNA is made in a prokaryotic cell and it's like dangling off, it can right away hook up with a small and a large ribosomal subunit and immediately start translating that. So you could be transcribing and translating literally simultaneously in a prokaryotic cell, where for us, we don't do that because we have like 30,000 genes, and depending on how we do our exon recombination, right, how we splice together our exons, we can get alternate protein products, right, just from a limited amount. And then what else did we learn? What's something that disrupts like the idea of the central dogma? What's something we have discussed most recently? So the end product may not be a protein. The end product may be some sort of RNA molecule, right, that helps regulate what proteins are made. So that could be an end product as well. Remember we said it goes DNA to RNA and proteins, but probably if we look at the evolution of the genetic code, probably what came first, DNA or RNA? RNA, because RNA could act as both an enzyme and as a keeper of information. And that brings us to, what is it? Big idea number 3, the way information is stored and transmitted and how you respond to it. Okay, so I just went over all the big ticket items there, and plus what we've kind of studied the whole year. What questions or concerns do you have, or would you like me to review anything in further? Yes?
I'm just, because I remember like there was a whole thing on it. Is water a covalent or a hydrogen?
Okay, so let's talk about that. So when you talk about water, okay, so if here's a water molecule, here's another water molecule. What kind of bond is that?
Polar covalent. It's polar covalent. Okay.
They are sharing a pair of electrons, but the oxygen shares it a little bit, hangs on to it. This right here, that is the hydrogen bond between two different ones, because this is slightly negative and this is slightly positive.
So it's holding two water
Yes.
molecules together. Okay.
Yes, or it's holding guanine to cytosine is a hydrogen bond. Okay, what else? Yes.
I just had a question. For the multiple choice section, how many questions is it going to be?
No, I haven't read it yet.
Do you want help?
2 or 3?
Oh, I just thought 1. 100 points just based on one question. I hope you get it. 100 or nothing. Yes.
It's like a weighted, like a percentage, correct? Or something?
No, I don't weight my grades.
Oh.
Points are points with me.
Oh.
Yeah, it's more points. The final is more points than a unit test. It's 200 points as opposed to 100 points.
So each part is 100. Does that mean like the weight is
Can I
100?
finish before you go in? Does that mean that the
Yes,
essay is
brothers.
100 and the multiple choice will be 100?
Uh-huh.
Thank you. So that makes me less stressed.
And we have a lab too, so that makes it better.
Yeah.
I love labs.
You love labs? You
Yeah.
go. You go. Okay, what else? Yes.
Can you give us an example of like a question that would be like connecting multiple?
Yes, I could say something like proteins play an important role in maintaining homeostasis. Describe the structure of a protein and relate its structure to its functionality in a cell membrane and how that would maintain homeostasis.
To its functionality in a protein.
Functionality in a cell membrane.
I can actually do that. Thank you. Got figured out.
Okay? Now, sometimes they give you choices. That used to be a lot in the past. They would give you choices. There are all kinds of exams released. Go look at them. And I have them too. I'll post all the ones that I can release. I'll post those and you can start looking at those. I'll work on that for you. Yes?
You would strongly recommend viewing the past essays, correct? That we've done this.
Yeah. Well, and the reason why those past essays are there, those are very much unit specific, right? But those check to see if you understand the material of that unit. And then I expect you to synthesize them together.
But should we know the detail as we did for each unit?
Always, you're never going to ask a teacher, should I know the detail? No, just forget it. You don't know what they're going to ask you. I am going to tell you, by and large, most students, what I hear again and again, there might be some questions they just flat out didn't know, but the biggest problem is being able to read it fast enough and respond. Because they'll look at the question and go, I know all those details. You didn't need to tell me that. You could have asked me that as a question. I would have appreciated that because I could have gotten it right. But instead, you wrote it in the paragraph beforehand. You saw some questions like that, though I still can't talk to you about it yet, in the test I just gave you, right? I wrote the long paragraph questions. You're like, yeah, I know what. And then the question I asked was like, meh. Okay? So it just took you a long time to get to maybe a very simple question, or it could take you a long time and then have a more complex one. But they'll give information along the way. So the best thing you can do is practice, time yourself to take questions. See how quickly you can move and still be effective in answering them. Yes?
Thank you.
Will the format be the same as like every test that we've taken, or
Yes.
is the essay gonna be okay?
Uh-huh.
Is the past highly suggested reading and thinking for the test?
For your multiple choice, it's that part I can let you off the hook on, okay? The details, except for the last unit, you need to do those highly suggested reading and thinking. The details, only reason I forced you to go through that and learn the details so you would actually read it and have to know it well enough that you could synthesize something. But I wanted to expose you with real life relatability to your content. But I'm not expecting you to memorize that particular detail of that particular example. The only ones I need you to know details are the things that we have over in our content. So that part I can let you off the hook for, except for the last unit.
So you had 5 or 4?
Oh, yeah, because I already asked you on these, right?
Yeah.
Yeah, so it would just be the last chapters that I haven't tested you on.
Okay.
Yeah, just the last chapters. So that would be 17 and 18, 19.
Yeah.
Yeah, just those. Those silly chapters.
No.
Yeah. What else? Yes.
There's gonna be essays from 17, 18, and 19, right? Yeah, I know they are. I don't need it.
Something else worthy of my time. No. Yes.
Will 17, 18, and 19 be in more detail than like the previous?
Oh.
Yeah. You would, because I won't have tested you on those in the multiple choice, so expect to see a lot of those on the final, which is a good thing for you actually. Because if I have more questions on one unit, that's to your advantage.
Yeah, do that.
Because you just went over it. What else? Okay. And then in your review section, if I haven't put it in there already on your, I don't know if it's in your resources, but there are practice AP questions already given, like, and answers to all of them. And some of them will be within these units we've discussed, and some of them not. I would take them because they're, you know, relating. I believe I broke them down when I first made them because I synthesized them from whatever release tests were out at the time since this most recent exam. When I put them by, you could like think of where it says unit 1, it's probably our first 2 units. You know what I mean? Where it says unit 1, it's like our unit 1 and 2, and unit 2 is our units 3 and 4. So if you pay attention to those, I broke them up by chapter if I could. I couldn't always because if they spanned more than one chapter. So I encourage you to go and look at those as well in the resources from the anchor page. What else? Okay, now, before our next review, which is tomorrow, 7th period, okay, what can you do? What can you look at? Because do you have any other homework due for me tomorrow? No, so you could look at something to help yourself out tonight, right?
Sure.
So what could you look at? Tell your bio buddy, where are you at? What could you look at between now and tomorrow?
I'm gonna check over the ones I know the least, I'm gonna do the notes, I'm gonna basically I'm gonna go over what I know the least, and then make it know what I know the least, what I know the most, and start learning what I know the least.
I'm just gonna go over you.
Now, if I got a test and there was a 96 on a test and I got an 88 and then I got a 71, what one am I going to need to focus on? 71. One of these was a partner test, right?
Yeah.
So that's not an authentic score for you necessarily unless it's right up in there with all your other scores. Okay, so keep that in mind, you know, on the, which one was the partner test, unit 3?
3.
Yeah, so you might want to spend a little bit more time there just to make sure you're okay.
Partner finals.
Okay, do you feel any comfort? Are you okay?
No.
No, no comfort?
I feel better.
I feel lower.
I feel like I knew nothing. I feel like I knew nothing about biology.
Well, I mean, look at how well you responded to these questions where we just jumped around. That's a good sign. That's a good sign.
I was like panic testing all of these people. It's like, I don't know anything about biology.
Okay, make good choices. Any other questions? Belle's gonna ring a
Will
minute.
tomorrow be, since we've covered everything today, like, I think will tomorrow be more, like, we
Tomorrow is unit 2.
Yeah, unit
But
2
like,
based.
today how we like, kind of talk about everything and somewhat unit 1, will tomorrow be like more unit 2?
Well,
or what?
unit 1 lent itself to that because of the introduction chapter. So I could do that. So whatever details you want to talk about. Again, you have specific chapter reviews already on YouTube, right? Go watch them. Okay. Make good choices.
It's a product. If you count